our speaker for this evening, coming to us live. Are you in New Haven, actually?
I am. I'm in my office.
Okay. From New Haven, Connecticut is Dr. Erika Edwards. Dr. Edwards is a professor of ecology and evolutionary biology, curator of botany at the Peabody Museum of Natural History, and the director of the Marsh Botanical Gardens, all at Yale University. Just a little background, trying to look at her. I can look at her here, but then I can't look at there. Erika received her undergraduate degree at Stanford and then her PhD at Yale. After her PhD, she was a postdoctoral researcher at the University of California, Santa Barbara for a couple of years. And then she was a professor at Brown University for 10 years before returning to Yale as a professor. She has published about 100 scientific papers, several of those in top journals like Science and the Proceedings of the National Academy of Sciences. Her research interests kind of run a broad range of plant evolution topics, including the evolution of C4 and CAM photosynthesis. If you are not a botanist or don't remember introductory biology, those are types of photosynthesis that are often seen in plants living in hot or dry environments. She is also interested in the evolution of leaf form and function in Viburnum, which if you're a gardener or a landscaper, you may be familiar with Viburnum. Among her many accolades, Erika is a recent past president of the best scientific society in the world, the Society of Systematic Biologists. And she is also a recent winner of the Presidential Award for Early Career Scientists and Engineers. I have to read that because it's a mouthful. The PCASE Awards, we know these as the PCASE Awards, and that is the US Presidential Award. So she was given that award in 2016 from President Obama. And I think that's it. I think that's all I have to say. So with no further ado, the title of tonight's presentation, you can see it. It is Darwin, the Modern Synthesis and 21st Century Evolutionary Biology. Let's give her a warm welcome.
Thank you. I think you just went mute, but maybe that's okay. Can you hear me okay?
Yeah.
Yes.
Okay. Well, so thank you so much, Andy, for the invitation to speak. It's such an honor, and thank to all of you for coming and listening tonight. As an evolutionary biologist, to be asked to sort of be a spokesperson for Charles Darwin is sort of one of the most humbling things that can happen to you. And so I want to start by just thinking about Darwin for a couple of minutes before we get into the meat of the science. And let's see if I can, oh, my slides won't move. Darwin was undoubtedly one of the best biologists that ever lived. I think most people would agree with that statement. And I would say it's an interesting time to be giving a talk in honor of one of our past heroes, because I would argue that my field, evolutionary biology, is going through a bit of a reckoning right now. We are full of hero worship. We love our ancestors, the people who put together evolutionary theory and initiated the work that we continue to build on. But what we've been realizing, some of us have realized later than others, that the history of evolutionary biology is troubling in a lot of ways, and that a lot of evolutionary theory has been used to support very racist ideologies and activities and politics. And some of our most esteemed heroes have dabbled in that over time. And so as we sit here on Darwin's 213th birthday, I think we can approach our admiration and celebration of Darwin in a few different ways. And I think one sort of frame of mind that I've taken on board lately is that we can sort of, greatly admire the past accomplishments of people without assuming that they are singular people in nature. In other words, Darwin was a brilliant person. He had a brilliant mind. He was an incredibly clear thinker. And he was also in the right place at the right time. And all of those things go together to sort of create discovery. And so it would be remiss for us to say if Charles Darwin never existed, we would have no theory of natural selection. Of course we would. It would just be put together by a different person in a different time and a different place. And so that's really the progress of science. That's how science works. It's a collaborative group effort. And we can celebrate what people did in the past while also sort of realizing that if it hadn't been them, it probably would have been someone else. And that's what sort of the discovery of truth is all about. It's inevitable and it will sort of emerge if people are working on it. And when I went to sort of the International Darwin Day to see what is this all about, I found this quote that I thought was pretty wonderful. And this is sort of, I think, the goal and the vibe of Darwin Day as we celebrate it. And the website says the mission of International Darwin Day is to inspire people throughout the globe to reflect and act on the principles of intellectual bravery, perpetual curiosity, scientific thinking, and hunger for truth, as embodied in Charles Darwin. So in that spirit, that's how I would like to celebrate Darwin Day with you today, celebrating scientific progress as a great collaborative effort. And when we go back and we look at Darwin as the person, what was special about him and his time and his place, he had a lot of opportunities to travel the world, more so than most people did. But what he brought to the table was an amazing ability to observe and to speculate. So Darwin, I would say, was a consummate, what we call naturalist, a natural historian. He was unsatiated in his desire to understand every detail of the organisms that he looked at. And one of my favorite quotes of his that isn't used as many as others, he says in 1857, "I am a firm believer that without speculation, there is no good and original observation." And this concept of speculation is so essential to scientific progress, and yet it's often thought of in the general public as something that you shouldn't do, and it's something that scientists don't do. And I'm here to tell you that scientists do it all the time, and the best speculators often make the best scientists. And Darwin was a no-holds-barred speculator. He took the observations that he made and he formulated grand hypotheses from them. And that's what we still do today. And the one other thing I want to say about Darwin and what was sort of special about his view of evolution and how he came to understand the concept of natural selection is that he was very much embedded in the world of ecology. So if you read his early writings, he was sort of not just charting out the basic theory of evolution, but he was also charting out basic principles of ecology. And in his world, ecology and evolution were completely entangled with each other. You wouldn't really be able to separate one from the other. And what's amazing about this is that the basic theories, the basic evolutionary principles that we still use today, they came out of this understanding of ecology. Darwin didn't even know what genes were. We had not discovered DNA. There was no sort of mechanism of inheritance that we understood at that time. And so the basic principles that then sort of led into evolutionary genetics and a lot of work on genes and genetic transfer of information from parent to child, that all came much, much later, long after Darwin had passed away. And in fact, there was this sort of second bloom of evolutionary theory in the history of science that was led by the likes of these folks here. And this was sort of in the sort of early to mid 20th century. And this period of time, it lasted maybe 12 or so years. And the whole time is called the modern synthesis. And what happened during this time is that Darwin's theories of natural selection were completely adopted, but there was no sort of mechanism. We had no understanding of how that would work. And what happened is that in around 1900 or so, this very old work by Gregor Mendel, who was a, oh my gosh, a religious man. I can't remember what they're called, right? A monk, sorry. He was a monk who was very interested in inheritance, and he had done a bunch of experiments with a bunch of different plants and actually discovered essentially what we call genes today, that there is sort of these discrete regions of genomes that are passed down and mixed during crosses, during reproduction, and given to ancestors at different ratios, and that those little discrete loci, these little things called genes, are actually responsible for the morphological variation that we see across organisms. That was a huge discovery, and it was rediscovered. And then this crew of people who are sort of the forebears of population genetics today went to work mathematically to try to understand how all of this worked. And so during this time, which was called the modern synthesis, I would say that evolutionary theory definitely shifted in terms of thinking about ecology and organisms and the diversity of forms that we see today, and shifted pretty narrowly into population genetics, or how populations can change through evolution from one generation to the next in small sort of steps. And an example of that is this, aptly named example of Darwin's finches. So when Darwin was traveling around the world as a naturalist on a boat for about 5 years, the Beagle, he made a stop in the Galapagos Islands. And this was an incredibly influential period of time for him, because he started noticing that the finches on the different islands in the Galapagos had, very drastically different beak morphology. So if you can, I gotta go to this screen, sorry. So here's a beak of a finch from the Galapagos, and they vary a lot in shape and size, right? So some can be very narrow, some can be very thick, they look really different. I'll show you a slide in a second. And so this sort of variation in beak sizes and shapes across a bunch of different species in the Galapagos was where he started to get this idea of natural selection, which is sort of demonstrated by this graph below. And the idea is that there is a wide variation of behavior of a trait in a given population. So you have a lot of a sort of standing variation between individuals and how you look or act in some sort of way, right? So there's a standing variation pool. And then what happens is that the species, or the individuals who have the variation in the traits that make them most successful in those environments, they will actually get the most resources, they'll be most fit, they'll outcompete all the other individuals, and they will be more likely to pass their genetics on to the next generation. And that whole process is called natural selection. And so this idea is that evolution is a very gradual process, and that you have continuous variation in a bunch of aspects of the organismal morphology, and those are selected on sort of gently, one generation to the next over time, until you get these little shifts in the population dimensions of that trait, right? And this is an example. This graph is a very famous graph of evolution in action, and it was one of the first examples that we had of evolution in action. So a pair of, sorry, I didn't mean to do that. A pair of scientists, Peter and Rosemary Grant, in the 70s, they returned to the Galapagos Islands specifically to study Darwin's finches, so the finches that inspired some of his theory. And what they found was that it was kind of lucky, which a lot of scientific breakthroughs are. But what they found was this was the distribution of beak sizes, beak depth, this line right here, in 1976. In this population of Darwin's finches on this one island of Daphne. And while they were studying this population, there was a very huge drought. So between 1976 and 1978, there was a very large drought on this island, and that changed the structure of the vegetation. And what they were able to do was sample the population in 1976 and then sample it again after the drought, where there was actually high mortality of finches, high mortality of lots of things. And what they showed was that actually there was a differential selection of finches for larger beaks. So the mean size of the beak had actually changed. And that is Darwin's theory of natural selection, right? So what this means is that the finches who had a greater beak depth were actually able to survive and reproduce more than the finches that had a smaller beak depth. And that shifted the morphology of that species. And that is sort of the paradigm of population genetics and evolution, and was for a long period of time. So let me go here. And this is just to give you a little more detail. The reason that this happened is that the drought actually changed quite a bit the reproductive strategies of the plants that were growing there. And it shifted the size of the seeds that were available for finches to eat. So in dry years, larger seeds are available, and in wet years, smaller seeds are available. So it was very directly correlated with the size of the beak finch, who could survive and who couldn't. And so I would argue that the modern synthesis was focused on understanding evolutionary mechanisms at this scale, at the population level. And to a large degree, another scale was entirely neglected, and that is the tree of life. So another big concept in Darwin's writings was the idea that there is a singular tree of life, that life evolved once on this earth, and that we are all connected to each other via this process of evolution by natural selection and sort of just having ancestors and descendants. So there's a continuous line of ancestors between you and the very first, molecule that we would say was alive. And it's between you and them, and it's between, my favorite Viburnum and that same ancestor. We share one common ancestor and we're connected in this way. And the idea of the tree of life, or the idea of using what we now call phylogeny to understand the evolution of organisms, was largely lacking at that stage. The field of systematics was there, but they didn't think that we would ever be able to sort of reconstruct evolutionary relationships between organisms well enough to do anything with that material. And so this is what we're missing when we just focus on population genetics. We are unable to get to the place where we can actually try to understand how the vast diversity of form and function that we see across the tree of life has evolved. For this, you need comparative biology, and for this, you need phylogenies. So my host, Frank Anderson, works on phylogenies. I work on phylogenies. This is a whole view of evolutionary biology that is sometimes termed macroevolution. I'm not a huge fan of that phrase, but we'll just talk about phylogenies and the tree of life today. And so what can they do for us? What they are is it's a sort of depiction of the diversification history of species across the whole globe and across time. And there's a lot of information in these phylogenies that we can use to understand how organisms evolve. And what's really kind of interesting, to me at least, is that this is a relatively new phenomenon for evolutionary biology. And in terms of when the sort of theory started with Darwin through this sort of dark period till we get to sort of the modern synthesis period in the 1930s and 40s, there was some really pretty decent development of phylogenetic theory during that time, but it was largely ignored in the modern synthesis and not really incorporated into it. And it wasn't until we get into maybe the 2000s that phylogenetics and reconstructing the evolutionary relationships of different species and using that to understand evolution became sort of commonplace as sort of just another major tool in evolutionary biology. And I'd argue now it's like sort of the major tool in evolutionary biology. And so what I want to give you a little hint of trying to understand what I mean by using phylogenies. We'll just go through a little cartoon before I jump into real phylogenies and sort of real world problems. So what do I mean by using phylogenies? So say you have all of these dots each dot represents a species. And the color of these dots represents some sort of character or trait that you're interested in understanding. So let's just, because I'm a botanist, call them flower color. So the red dots are red flowers and the white dots are species with white flowers. And when you just have a pile of species like this, it's really, you can't make any inferences about which flower type evolved from which, how many times each flower type evolved or what. You don't know anything about the evolutionary history, right? You just know that there's 3 species with red flowers and a handful with white. So when you actually start to build the evolutionary relationships between them, and these days we use molecular gene sequences to do that, it's become very sophisticated and pretty good, I think, the methodologies. What you find in this case, so this would be a phylogeny, and this depiction represents each one of these sort of, oh gosh, I'm on the wrong, sorry. Each one of these little divergence events represents an ancestral speciation, right? And so you can see who's more closely related to each other and who's more distantly related to each other. And all the tips are living taxa that exist today. So this is really kind of the family tree of these organisms. And once you have that, then you can start to make all sorts of inferences about character evolution. For instance, I can say, it looks like red flowers evolved once here, and then they evolved once over here. And it looks very likely that the ancestor of this group had white flowers, right? And you can start to sort of, add other layers onto this phylogeny and sort of use it as a scaffold to bring together a whole bunch of information that helps you sort of explain the evolutionary history. So for instance, say I have more data about these species and I know who's pollinating them. So say if you are a yellow dot, you're pollinated by a hummingbird. And if you're a brown dot, you're pollinated by a bee. Well, what we can say here is that it looks like when I'm over in this clade, every time I evolve a red flower, I shift to having a hummingbird pollinator. But when I go over here, I have hummingbird pollinators, but I don't have any red flowers. So what's going on? So I would say on one level, it seems that red flowers are associated with hummingbird pollination, but not always. Why? And so what's special about this clade? Or what's special about this clade? What makes them different? And you could also start to look for, well, what are some mechanisms that might allow an organism to evolve red flowers? What is required? And maybe you discover that actually there's some sort of biochemical precursor that you need to have in order to make a red pigment. And you look and you find that everyone in this clade has that precursor. Nobody in this clade does. Furthermore, you can actually use this tree to sort of date the divergence events across time using all sorts of calibrations that I won't get into. And so you can get a timeline of the order of events that are happening. And so you can say, in some sense already, just from having a phylogeny and a little bit of extra trait information, you've come up with a very interesting and valid evolutionary hypothesis, which is that at some point in time, between 10 and 5 million years ago, say, this clade evolved this biochemical precursor trait that allowed it to make red pigments. And because it can make red pigments, anytime it makes a red pigment, it switches pollinators. So there's some link between being hummingbird pollinated and being red flowered in this group. And so that's what I mean when I talk about using phylogenies. And that's sort of the bread and butter of my group. That's what we do. And it's a really exciting time in the world to do that, because I'd argue that that sort of forgotten part of the modern synthesis, the phylogeny, the tree of life, that area has been exploding over especially the last 20 years. And so when we look back and see where we've come as we sort of enter the 21st century, I would say that phylogenetics has been going through an absolute frenzy, both of sort of data accumulation. So we now have massive amounts of genetic data that we can use to build phylogenies. And we've also been sort of transforming the way we do things. So there's been a ton of methods development, theory, and implementation of new theory into new methods that allow us to build bigger trees, better trees, and also sort of analyze a whole bunch of organismal data onto these trees. So it's been really, really fun. It's been a fun time to be involved in this. The other thing that I would say that's happened over the last 20 or so years is that phylogenetics has also sort of looked outward, in that you can actually apply this tree of life information and knowledge to all sorts of subsets of biology and even sort of societal problems, planetary problems that we're dealing with now. So phylogenetics has made its way into things such as conservation planning. So instead of just thinking about species richness of certain areas, we now think about the phylogenetic diversity of certain areas. So how much of the tree of life are you capturing if you want to preserve a certain piece of land? And is that more important than preserving, say, 10 species of the same genus, right? And then, of course, more recently, I put this up at the beginning, but just to sort of emphasize how phylogeny is everywhere now. This, of course, is a wonderful website that I encourage everyone to go to if you're interested in understanding the evolution of COVID-19 strains. These are all generated from sequenced genomes of our COVID-19 virus as it's been circulating around the globe. And this is how we know, for instance, when Omicron sort of emerged on the scene back in, I guess, late November, early December, that it was not a variant of Delta. We knew that because we built a phylogeny of these sequences. And so you can see here in this big blue clade, this is the Delta clade. They're all each other's closest relatives. And Omicron came from deep in the tree down here. So it had been sitting isolated for a long period of time and then just exploded onto the scene. And so this kind of approach is very important in evolutionary medicine, human health, conservation, et cetera. I want to turn back to the science now and just ask the question, if we're living in this world of, big data, fancy data, fancy informatics, all that sort of stuff, where is the place for old-fashioned natural history? So if we go all the way back to Darwin as the sort of keen observer that he was, he came up with all of his ideas and theories simply by sort of walking around in forests and in other landscapes with his notebook and looking at stuff, right? And, there's an immense value in simply just looking at stuff. And so are we still doing that today? Are we still just kind of checking things out and observing things closely? And I would say we definitely are. So today is a wonderful time in the world to be a natural historian. If you want to still call yourself a naturalist, you've got a lot of tools that you can use to look at organisms. So you can look at them the old-fashioned way, and there's still a lot of information. This is a sheet of an herbarium. This is an herbarium sheet. This is how we sort of process plant specimens and store them in perpetuity. And they contain a lot of information, but you can also look at organisms however you want to. We have an amazing ability to look at sort of the microanatomy of living tissues now with all of this sort of CT scanning that we can do. We have immense opportunity to look at any sort of comparative genomic elements you want. We're currently right now involved in a sort of spatial gene expression analysis in a portulaca leaf. So you can take a slice of a leaf, preserve all the RNA that's being made at a given point in time, put little tags on it so they're spatially located, sequence it, and then get a photograph of gene expression back out the other side so you can see where everything's happening inside a leaf. It's kind of just an amazing time to be alive. And so that's kind of where we are in our world in terms of how lucky we feel to have all of these approaches open to us as we explore the clades that we love. And the approach that we've developed, I'd say over the last 20 years or so, is an approach we call sort of the generation of model clades. And so what is a model clade? A clade and a lineage are kind of interchangeable words in my brain. A clade is sort of a monophyletic group of organisms. And just as in the world of genetics, they have sort of developed what they call model organisms, which are things like fruit fly, Arabidopsis. There's a little worm that everybody uses. These are individual, these are single species that thousands of people work on as a sort of genetic system, because genetics is so complicated that they need as much information and context as they can when they're sort of evaluating any one of their studies. And so genetics has gone forward very quickly because they've sort of adopted this model organism idea, like let's all work on fruit flies until we understand fruit flies. And I'd argue, and my colleagues would argue, that at the macro scale, macroevolutionary patterns are no less complicated or difficult to understand than our genetics. And if we really want a holistic understanding of organismal evolution at that scale, we should have model clades. We should have clades where a lot of people work on these clades from as many different angles as possible. And so we've been trying to develop 2 model clades in the lab, and hopefully have lots of people sort of jump aboard and sort of take them on as their own as well. This one here is called the Portulacini, and it includes things like cacti. And that's where we do a lot of our work on C4 and CAM evolution, as Andy mentioned earlier. I'm not going to talk about this today. Instead, I'm going to focus on Viburnum. Viburnum is a wonderful group of plants, and we have been studying them. Well, I myself have been studying them for about 15 years or so. And my collaborator, Michael Donahue here, actually, he's about to retire, I think in June. But he did his PhD dissertation back in 1980 on Viburnum. So he's been studying it for a very long time as well. And this is a group, it seems sort of inconsequential at first. So it's not a large group of plants. It's about 165 species. They're all woody. They're shrubs and trees, kind of nondescript, if I have to admit it. They live mostly in the Northern Hemisphere, but there's a really interesting distribution in Southeast Asia, and they're hyper diverse in China. And then another really interesting little radiation down into the Andes of South America, and we'll talk about that at the end. But really, they're thought of as a relatively straightforward Northern Hemisphere temperate woody shrub and tree group. And this is the general, this is outdated. There's even more people working on it now, but just to sort of emphasize that model clades require lots of people. And so there have been lots of people that have been involved in this work over the years. And this is Michael and me, and we sort of have been sort of leading the charge of let's get everyone on board with this group. And let's look at everything that we can about them. So one of the ideas of a model clade approach to evolutionary biology is that organisms are incredibly complex. Lots of things are connected that you might not know are connected. So if you're really interested in understanding the leaf evolution, you should also know a lot about the stem, and you should probably also know about the roots, and you should probably also know about the flowers, because there's a lot of modularity in organisms, but those things also are connected to each other. And so if you want to understand how one thing changes, you should have a good understanding of the rest of the organism in order to do that. It's that context thing again. But today, what I'm going to do is really just focus on leaves, partly because leaves are fun and pretty to look at, and everybody in the room or on the Zoom has looked at a leaf, right? You might not have looked at xylem in a stem, but you've certainly looked at a leaf before. And one of the things that's really fun about Viburnum, they're not super diverse in terms of their floral morphology, but they're really diverse in terms of their leaf morphology. So if you look at these examples, these are all different species of Viburnum. And if you have spent much time sort of in temperate deciduous forests, you might recognize a lot of these leaf types. For instance, this looks kind of like an oak. This looks kind of like a maple. This looks kind of like a beech, right? But they're not, they're all Viburnum. So Viburnum sort of converged on a lot of sort of different kinds of leaf shapes over the course of its evolution. And so the two like short little stories I want to tell you today. One is sort of older. We worked on this maybe 5-ish years ago, which is concerning how when Viburnum moves across the globe, when it's making these tropical temperate transitions and things, what happens to its leaf shape? How does that change? And then I'll end the talk on stuff we're doing just right now, kind of right in the middle of it, where we look at actually one little subclade of Viburnum as it's moving through an archipelago, what we call, of cloud forests in the Andes Mountains. What's happening to its leaf shape as it's doing that? So we'll start first broadly with the biome one. And so this has been ongoing. We have been slowly, steadily collecting a phylogeny of Viburnum and adding species to it sort of painstakingly over many years. And I would say now we are very close to a complete phylogeny. And what I mean by that is that we've basically sampled every living species with just a very handful of exceptions of pretty rare species. And for a group of 165 species or so, that's not super common. So it's really nice to have such a sort of complete sampling of the clade as they're alive right now. And we've been working on dating the origin of Viburnum. How old is it? And also sort of when some major transition events were occurring in the group. And in this first image, this is a phylogeny, and don't worry about not being able to see anything. It's okay. This is just a Viburnum phylogeny. And we think that it's a relatively old group. It's been around for maybe 70 or so million years. And we think it started in, ironically, since it's mostly a northern temperate group, we think it actually, its origin was in these warm forests of Asia and Southeast Asia. So when I say warm, it's not necessarily a pure tropical forest. There are these sort of what they're called lucidophilous forests, warm, mildly seasonal forests that are mostly evergreen, not tropical, sort of subtropical, but they definitely don't freeze. And we think that's the most likely sort of scenario for the origin of Viburnum. And what's kind of cool about Viburnum is that most of the species diversity now is in the temperate zone. And we think there have been multiple transitions into cold temperate forests across the group over the years. And this little group is the group we'll get back to at the end of the talk. This is one radiation in Central and South America out from a temperate lineage, it back down into, so coming out of Eastern North America and going back down into Mexico and Central and South America, back sort of into more tropical-like environments. Okay, and so when we look at leaf shape and size and phenology, whether a plant is evergreen, so it has its leaves on all the time, or whether it's deciduous, like the forests around here where they drop their leaves for the winter, we find that just leaf form and phenology are perfectly correlated with each other, and they're also perfectly correlated with these biome shifts. So when you look across the whole group and you see lots of warm subtropical to cold temperate shifts, you see corresponding changes in those species, just like the red and yellow dots that I was showing you in that cartoon, corresponding changes in leaf form and function. And that when you look across the whole tree, what you find is that these very consistent leaf syndromes emerge. So when you're in the tropics or subtropics or even these montane forests, you tend to look like this is a Viburnum, where you're sort of evergreen. You never sort of drop all your leaves at once. And you have leaf shapes that are very elongate, and they have very simple margins. So they're very smooth around the edges. When you go to the temperate zone, you immediately become deciduous. So you go through a period of leaflessness in the wintertime, and your leaves become more complex. So the shape of them becomes different. They're rounded. They often have teeth around the margins, and sometimes they're lobed. And we think a lot of these transitions happened in Asia, where there is the highest diversity of Viburnum species. And we see lots of different parts of the clade highlighted in here. Here's one element of it. Here's another clade, subclade, where you see within these groups clear transitions as you move north. These leaves are situated on sort of the midpoint of the range of each of these species, where in the tropics, you have these tropical looking leaves, and in the temperate zone, you get this very consistent and recognizable leaf shape. And when you think about it, and you think about when I had a small child, we were doing this work, and I would read this storybook to him and marvel at this actual page, because I was just imagining all my viburnum leaves falling down across this bunny. But this is a really wonderful depiction of what the leaves of northern temperate forests look like, right? You can recognize a lot of the leaves that you see around Illinois. And viburnum leaves fall directly in here. So there is a general trend when you go into these deciduous forests of the emergence of really complex leaf shapes. And this has been known for a very long time. So this paper is over 100 years old by Bailey and Sinnott. And these were sort of two ecologists who actually just made a comparison between the trees living in a lowland tropical forest, in this case, Brazil, and the trees living in the cold temperate Eastern North American forests, right? And they found an amazing difference in the percent of trees that had entire margins versus these complex margins. So 90% of the trees in Brazil look like this, whereas 90% of the trees in Eastern North America look like this. It's almost a complete flip-flop. And people have actually gone through and sort of quantified this over the years. And they can find, actually, it's pretty strange to me that there's a sort of almost linear relationship, right, between mean annual temperature of a given environment and the percent of species that have an entire margin versus a toothy or lobed margin. So this is a very general trend across the world. And people have looked, it's very strong in going from sort of North and South America, but when you add other continents, it gets a little messier, but it still holds basically. So there does seem to be some sort of fundamental thing going on with leaf shape and temperature. And it was what we recovered in Viburnum as well. So now we've seen it sort of in action operating across a clade. And so people have been puzzling about this for a long time. And most of the explanations that have been proposed for this pattern have focused on the ecophysiology or the function of mature leaves and how they perform gas exchange. So leaves are a very important part of the plant because they're the place where carbon dioxide is taken up to make sugars. And it's also the place where they lose a lot of water to the atmosphere in exchange for that CO2. And so there's a lot of ways in which the shape of the leaf itself could influence those dynamics. And so that's where a lot of the attention has been focused. However, most of the explanations don't make a ton of sense. So it's been a bit of an enigma. And what we were thinking when we were seeing this in Viburnum is that no one is really focused on this other aspect that is also perfectly correlated. And that is this transition in phenology and the rhythm of growth. And so when you're in the tropics and you're in these warm climates, you look like this, but you're also evergreen, right? So you're making leaves maybe one at a time. It's just, and they stay for a long time. And when you're in the temperate zone, you're always deciduous. And what this means is that, and you can go out and look around tonight or tomorrow at what's going on in the trees surrounding you that don't have any leaves on them. They have these buds, they have these overwintering buds at the ends of their stems. And those buds are full of leaf primordia. They are full of the next leaves that are ready to go for the spring. And that's a really big element of what it means to be a temperate deciduous plant. And while we were working on this problem, this amazingly strange and interesting paper came out onto the scene. And it was from a handful of mathematicians in Paris. And they were interested in how things are packed inside leaf buds and whether or not that had anything to do with, they could explain via the process, via the theory of kirigami, if they could explain the sort of eventual mature shape of the adult leaf via the way it is folded and packed inside the bud. And they found that they could for a number of species. So this is, up here is a beech. And is this a beech? I guess that's also a beech. Yeah, this is also a beech. And so what they did was sort of create this model. So kirigami is like origami with one single cut. And it's, I guess, mathematically proves that you can make any two-dimensional shape this way. And so what they found is that the way that these things were folded inside the buds completely predicted the mature leaf shape once the leaf had emerged from the bud and fully expanded. And while that sort of blew our minds, we were also noticing some other things, and that in our temperate species of many viburnums, here's 2 different species, what we found is that the first leaves that were formed in the spring, this one here and this one here, look very much like a classic temperate leaf. And those were the leaves that were formed inside the bud. Usually they make usually 2 pairs of leaves. So these 2 here. In some species, they actually continue to produce leaves. We call them neoformed or free-formed leaves as the branch grows during the season. And whenever they did that, they reverted to a much more tropical leaf shape right here. And so we were thinking, well, maybe this leaf shape doesn't really have anything to do with mature leaf function necessarily. Maybe it has something to do with the way these things are packed inside these buds. And so the new sort of idea is that this temperate leaf type emerges not because it's ecophysiologically advantageous for gas exchange, but because it is resulting from packing your leaf primordia in buds really tightly so that you're ready to go in the spring and sort of emerge and get your leaves out there ready to go. And so we started then getting really interested in bud morphology and what's going on inside buds. And when you look at what's going on in tropical versus temperate viburnum buds, you see something really interesting is that there's very little leaf formation going on inside tropical resting buds of viburnum. So basically, it was hard to find resting buds in some of these species. Here's one where we kind of pulled these bud scales off, and you can see there's just a little nub of tissue. And what these species tend to do is they still go through a resting phase where they make bud scales and they put it on, but they don't really grow there. And then when they start sort of emerging and growing again, all of this is happening outside of buds. And so even at this stage, there's very little leaf tissue that's even been made. In fact, this is mostly the petiole and then the back vein. And so there's not a lot of sort of leaf blade that exists inside these buds. And when you get to the temperate zone, of course, it's a very different story. So this is Viburnum dentatum or arrowwood just emerging in the spring. And you can just see, these are the 2 bud scales here. And then this is all the leaf tissue that's been packed in there over the winter, ready to go. So it's a very different scenario. And what we've been able to do is use, the sort of 21st century abilities to observe using these micro CT scanning so we don't have to destructively disrupt buds to see what's going on inside all of them. And this is still sort of a work in progress, actually, because you get these CT scans and then you have to figure out how to sort of actually in silico sort of dissect the leaf tissue from the blade tissue. And it's non-trivial, and I'll skip over this. But when you actually do the analyses, what you find is that what we sort of casually observed is true, is that in these entire margins, tropical-like leaves, you have almost no leaf tissue inside your resting buds. And that is not true when you get to the temperate zone. It's much more significant. And so we think this could be a new mechanism to investigate in other lineages. And as to leave this section of the talk, I just want to briefly show you this picture because I think a lot of people recognize the fruits on this plant, right? Yeah, it's a maple, but it's a tropical maple. In other words, a lot of the temperate lineages that we're used to, that we see around here with teeth and lobes and things like that, when you get down into these lucidophilus warm temperate forests where we see viburnum as well, they look like this. And so this would be a really cool study to see how many different lineages you can trace to this sort of situation. Okay, so I'm running out of time, but this will be quick. This is just another sort of example of, I would say, modern day evolutionary biology, natural history. And this is work that we're just doing now. It's still ongoing, and it's been an enormous amount of fun. So we have gotten very interested, if I go back to this little chart here, in this light blue clade, right? So this is a subgroup of viburnum that we call the Arvenitinus. And we think roughly maybe 9-ish million years ago, they migrated down from North America and actually started moving through Central and South America just over the last 10 million years. I think they only got to South America a few million years ago. And they are sort of restricted to these cloud forest environments. And so what's pretty neat about this is that you end up, if you look across the whole region, you get these very disjunct areas, right, that are separated from one another. And so they act almost as like sky islands or islands of habitat that is suitable for these viburnums. So they've been hopping along these sort of islands in the cloud forest, speciating over the last 10 million years. And we've done a lot of collecting all across this region. All these black points are places where we've gone, and we supplemented our own collecting with collections of different herbarium specimens. And what we've now been able to do is sort of resolve a really nice phylogeny of this radiation. So this is a pretty recent radiation as opposed to the broader viburnum. And what's kind of cool is that there is a clear phylogenetic, geographic pattern to their movement. In other words, it's undoubtedly so that viburnum has moved from the north down through the south. And so the most recent speciation events are happening down here in Bolivia. And we started working on this in Mexico. And one of the reasons we wanted to start working on it in Mexico is that a very long time ago, Michael Donahue worked on this for his PhD, and he noticed that there's like these two distinct leaf types in any of these regions that he goes. So if you go to Chiapas, a disjunct little chunk of cloud forest in Mexico, you get one species that has these giant leaves that are really hairy and really toothy. And then you get this other species here that has really, really tiny leaves, totally glabrous, no hairs on it at all, and no teeth. And if you go to Oaxaca, you find the same exact thing, right? Which is really nuts. So they're very different looking. And you can use a phylogeny to sort of investigate, alternative hypotheses for how that happened. So one hypothesis is that there are sort of two main clades of viburnum that are sort of co-radiating together, sort of in parallel through these regions. And one of them is a large, hairy, toothy clade, and one is a very small little, entire margin smooth clade. And that was the hypothesis that he favored back in 1980 or whenever he was looking at it. The alternative hypothesis, of course, is that actually you have sort of one main lineage radiating through, and every time it gets to a new mountain area, it diversifies into these two leaf types. Now that seems fairly unlikely, right? It's definitely not parsimonious. How could it be that you would kind of repeatedly evolve these leaf types over and over? So this is how we sort of started this project. That was our major question. And we've been working for years just collecting these plants and getting to know the clade, because it's probably not a very well collected clade. And we've worked across the region, and what we found is that as I said, there are these very distinct areas of endemism. And what I mean by that is that in these regions, there's multiple regions we've identified where there's a group of species that sort of live there together and they don't live anywhere else. And the same for this group and this group and this group. So there's a lot of geographic sort of stratification of the species in this clade. And when you go to each of these species, again, it's not just Oaxaca and Chiapas, it's throughout the whole radiation. You find a repeated set of leaf types that occupy these zones. And as we've been collecting this and studying it further, we found that there aren't just 2 leaf types as we started. We've discovered far more species that we knew existed and far more leaf types, and that there's really 4 main leaf types. There's this type over here that we're calling the dentatum type, and this is the ancestral type. This is the type that is present in Eastern North America, and it still exists in central Mexico and in Oaxaca, but then it kind of quickly dies out and is no longer part of this radiation. And for the rest of the region, you've got these 3 main leaf types here. You've got the big, hairy, toothy one that I showed you earlier. You've got the tiny little glabrous one. And then we have this 3rd one that's very sort of tropically looking, we call it. It's like very much our tropical tree species, where the leaves are larger than this one, but they're also completely smooth and glabrous and don't have any hairs on them. And when you actually do sort of the morphometric analysis of different traits, you find that they do sort of sort out into these kind of 4 quadrants of leaf types. And when you look at them across the tree, this is the big exciting thing that we've discovered, is that it is not that you have these 3 kind of lineages that have 1 leaf type, 1 leaf type, 1 leaf type sort of parallel radiating with each other. It's 1 lineage that's radiating from north to south. And in each region that it lands, it repeatedly diversifies into these leaf forms. And so this is what biologists call a replicated radiation. So it's when you get the repeated evolution of a set of things across space and time. And the most famous one of these, of course, are the anolis lizards in the Caribbean. And I don't know if anyone has heard this famous story, but this is the example of these things called ecomorphs, where these lizards have diversified ecologically into these little microhabitats, and they've done it over and over again in these different islands of the Caribbean. So it's a good classic animal example. Here's another animal example. This one's in spiders in Hawaii. And we're not sure. We're looking through the literature, but we can't seem to find a textbook sort of concrete example of this happening in plants up until now. So we're sort of presenting this, and we're very excited that we have almost like a brand new system in which to investigate convergent evolution of leaf types and what they could possibly mean. And so I'll end by saying that we're working on this. This is just, we're just sort of beginning to explore both functionally and ecologically what's going on. And part of the problem is that they all live very close to each other in these cloud forests. So it's not like one's in the desert and one's in the rainforest. They're all living in these cloud forests, and the environments to us seem fairly similar. So one thing we have done is set up these little sort of microclimate stations to get really specific data on those sort of very local environments of where these species are living. And just focusing on 3 species in Chiapas, these guys over here, I'll show you just a tiny bit of data. This is what the stations look like. So we just kind of go to these populations and we set up a temperature and humidity sensor, soil moisture sensors, and most importantly, this thing called a leaf wetness sensor, which just kind of sits there and measures what proportion of the day there's actually sort of air moisture. In other words, a cloud. These are cloud forests, right? So the clouds roll in and the world is wet for a period of day, and then they roll out and then it's dry. And so we've been keeping track of that. And in a nutshell, what we found is that there is significant but subtle differentiation of these ecotypes into their local environments. So this tropically looking guy here, this is the lowest elevation one, and it seems to live in generally warmer environments that actually are a little less seasonal. This one, our little leafed guy, tends to live in slightly drier environments, and also environments that are more open, so like less of a canopy forest, and have sort of very significant diurnal and seasonal fluctuations in temperature. So it can be hottest and it can be coldest. So it kind of deals with temperature extremes. And then the jucundum, this big hairy leafy guy, this is the one that lives in the cloudiest of the cloud forests. So it's at slightly higher elevations. There are different sort of aspects whereby the clouds come in and linger longer. So it's dealing with cooler and wetter environments. And so that's just the very beginning of trying to understand what these ecomorphs mean. And so I'm going to end, sorry, I took a long time. I'm going to end by emphasizing a couple of things about, I think, what it means to be a naturalist, and what I think Darwin probably thought about a lot of the time is that a lot of the time you're in the field and you're trying to make these observations and put things together. And a lot of the time, nothing makes sense, right? And so you're scratching your head a lot. What does it all mean? And when we're in the field, we have this joke of Mr. Natural. I don't know if that's way too old for most people in the audience. This was like a cartoon that was around like in the 60s. Mr. Natural, what does it all mean? Don't mean she. We talk about this a lot. But this is a phase that you have to go through, and it brings back the ideas of observation and speculation to me. I'm going to skip over this because I don't have time, but we did, just from scratching our heads in the field, discover new things that we think could really help us understand the mechanisms as to how you can get so many transitions between these leaf types so easily and so many times. And it has to do with developmental trajectories and maintaining juvenile characteristics in adulthood, because we were able to observe seedlings in the field. But at the end of the day, I just want to return to this, what I think is an important element of Darwin's worldview, which is that you have to be a good speculator and a good observer to be a good scientist. And the more speculation you do, the better observer you will become. So don't be afraid to be confused. If there's budding scientists in the audience, I don't know. But this is a period that often happens during your PhD. You go through a period where you're very confused about everything. And I'm just here to tell you that that is a natural progression of discovery. And so you should embrace it and not worry about it. And it's actually one of the most beautiful parts, I think, about being a scientist. And I think Charles Darwin probably would have agreed. So I'll end, but first want to sort of reiterate my thanks to all of my collaborators, past and present. I've been so lucky to work with the most wonderful people in the world. And science is a collective activity and a progress that only works through collaboration and sharing. And I'll also end on that note with just a shout out to all the people who have come through my lab over the years. This is actually a very out-of-date photograph. I've been so busy. I haven't been able to cut and paste like a bunch of new people into here. So I have to add this, but this is an ever-growing sort of group of people who I love dearly and who make my job a lot of fun. And so with that, I will, hopefully there's still people around who want to ask some questions. And thank you again for listening. And thanks so much for the invitation. It's such an honor to give a Darwin Day talk. So thank you.